It is my pleasure to be here at this 2020 FeSBE virtual meeting. I'm 𝑬𝒍𝒂 from the School of Pharmaceutical Science in Ribeiro, but also currently enrolled in Aarhus University in Denmark, and I will be the moderator of this symposium together with Professor Henning Ulrich from the Chemistry Institute in the University of São Paulo. And we will have talks by 4 excellent young investigators that will talk about the different aspects of purinergic signaling in neuronal or neural disorders. The first speaker, Deidiane, and Deidiane will talk. Deidiane Elisa Ribeiro, she's currently at the Institute of Chemistry in the University of São Paulo. She will talk about targeting 𝑃2X7 receptor in stress and depression. And Deidiane has developed her master and PhD in the Department of Pharmacology in School of Medicine of Ribeirão Preto under my supervision. And after that, and part of her PhD, she did also in Aarhus University as part of a double degree agreement between these 2 universities. And following her PhD, then she went on in this investigation with 𝑃2X7 receptor signaling and depression. And currently, she's working under the supervision of Professor Henning Ulrich, working at the University of São Paulo with the interest to unravel new aspects of purinergic signaling in animal models of Alzheimer's disease. So now I would like to invite Deidiane to present her data.
Thanks, Samia, for the kind presentation. And thanks, FeSBE organizers, for the opportunity to be here today. It's a great pleasure to discuss my PhD results regarding the 𝑃2X7 receptor signaling in stress and depression. So I will initiate my lecture talking a little bit about depression. Currently, depressive disorders affect more than 322 million people worldwide. According to the 50th edition of the Diagnostic and Statistical Manual of Mental Disorders, the major depressive disorder is characterized by the presence of 5 or more of the following symptoms during at least 2 weeks. Mandatorily, patients must present depressed mood and anhedonia, plus loss or gain of weight, decrease or increase in appetite, insomnia or hypersomnia, psychomotor agitation or retardation, fatigue, feelings of worthless or inappropriate guilt, diminished ability to think or concentrate, recurrent thoughts of death or suicide. Such a variety of symptoms states the huge complexity of the major depressive disorder, a mental disease that can be triggered by environmental factors, mainly stress exposure, genetic, epigenetic factors, and also personality traits. These vulnerability factors lead to neuroendocrine imbalance, neurochemical alterations, and neuroplasticity impairment. In this context, alterations in the HPA axis can result on high glycocorticoid levels, which leads to the impairment of the monoaminergic neurotransmission, increase in the glutamatergic neurotransmission, and activation of neuroimmune response. These systems interact with each other, and it will result in the neuroplastic impairment, include the decrease of neurotrophic factors released, such as BDNF, diminished the neurogenesis and apoptogenesis, dendritic atrophy, and glial cells dysfunction. So, looking for this figure, we can imagine the difficulty of finding an effective antidepressant treatment, especially because almost clinical available antidepressants have a similar mechanism of action, which are based on the facilitation of monoaminergic neurotransmission. As can observed in the timeline, we first had the monoamine oxidase inhibitors, then we have the serotonin antagonists and uptake inhibitors, We also have the selective serotonin reuptake inhibitors and the serotonin neuropinephrine reuptake inhibitors. Given the similar mechanism of action of these drugs, it's not surprising that the antidepressants share some features. Notably, most of them need of about 8 weeks of treatment to reduce the depressive symptoms by half. Moreover, even after this period, about 33% of patients do not respond to the antidepressant treatment initially prescribed. Therefore, a more effective treatment for depression is required. And the P2X7 receptors have been indicated as a possible target, since it is involved in processes that are dysfunctioning in both stress response and depression, such as the neuroimmune response, neurogenesis, glutamate release, and neurotrophic factors release. The P2X7 receptors are ATP-gated ion channels permeable to potassium efflux, as well as sodium and calcium entrance. The sustained activation of these receptors induces the formation of large pores, enabling the passage of molecules of up to 900 Daltons, such as NMDG and the ATP itself. The passage of these molecules may occur by two distinctive mechanisms: through the dilatation of the ion channel pore, or through the interaction of the receptor with membrane proteins, such as the pannexina 1. The P2X7 receptors are largely expressed in the central nervous system, mainly in microglia and astrocytes. However, its expression in the terminal nerves have been highly discussed. So it is proposed that stress exposure elicits a massive ATP, represented here in purple, and glutamate, represented here in orange, release with a consequent activation of the P2X7 receptors by ATP and NMDA receptors by glutamate. Stimulation of these receptors leads to enhanced potassium efflux, result in the NLRP3 inflammasome activation. Consequently, we have the release of interleukin 1 beta 18 and also TNF-alpha. The stimulation of these receptors can also increase calcium influx, leading to ATP release and glutamate release from astrocytes and neurons. Oops. Moreover, we have that the activation of NMDA receptor and calcium entrance can also lead to the NOS activation and consequent increase in the nitric oxide formation. So, in this context, reactive oxygen species production is also stimulated, and altogether we will have a neuronal damage. So, under stress, high levels of ATP, glutamate, pro-inflammatory cytokines are maintained by a regenerative circuit, which leads to diminished BDNF levels, decreased synaptogenesis, neurogenesis, and damage of brain circuits important for emotional regulation. Interestingly, a P2X7 receptor polymorphism have been related to the severity of depressive episodes. In this study, the presence of the GLL was investigated. It consists in a polymorphism that leads to the change of a glycine by adenine amino acid in the 460th position, and it results in the loss of function of these receptors. As can be observed in the graph here, the presence of the GLL is associated with a higher score in the hospital anxiety and depression scale in patients with both major depressive disorder and bipolar disorder. So, according to evidence, we have that 𝑃2X7 receptor signaling may be modulated by stress, and alteration in the function of these receptors are associated with the severity of depressive episodes. Based on that, we can raise the question, what's the effect of the antidepressant treatment of 𝑃2X7 receptor expression? So it was studied by me and collaborators in this work published last year. In this work, male oyster rats weighted in between 250 and 280 grams were used and exposed to the learned helplessness model. This model was chosen because of the antidepressant response can be observed just after repeated treatment, such as in the clinical scenario. This is important since I intended to investigate alterations in protease expression, and maybe it requires more than a single administration to occur. Then the protocol used consists in a pretest session where animals are exposed to 40 inescapable food shocks. And 7 days later, these animals were submitted to the same context, but now 30 escapable shocks were applied. In the test sections, the animals could escape from the shocks by crossing to the other side of the shuttle box. However, after the uncontrollable and unpredictable stress of the pretest session, the animals cannot learn how to escape from the shocks. This behavior is called learned helplessness and can be inhibited by antidepressant treatment during more than 1 day. And for this reason, the parameter analyzed in this model is the number of failures in the shuttle box test session. So during the 7 days of interval, the animals were treated with daily injections of 𝑧𝒂𝒏𝒂 or 6 injections of 𝑧𝒂𝒏𝒂 followed by a simple injection of imipramine, or another group were treated with 7 injections of imipramine. The next step was to investigate the alterations in 𝑃2X receptor expression induced by antidepressant treatment. And for that, animals were exposed to the learned helplessness pretest session as previously described. The same treatment protocol were applied, and 1 hour after the last injection, the animals was decapitated and had dorsal and ventral hippocampus as well as frontal cortex dissected for Western blotting analysis of the 𝑃2X7 and 𝑃2X4 receptors. As expected, here we can observe that the repeated but not acute treatment with imipramine decreased the number of escape failures, here shown in the black columns, but induced no alterations in the number of intertrial crossings, here in red, in the learned helplessness model, which can be interpreted as an antidepressant-like effect. Regarding the 𝑃2X7 and the 𝑃2X4 receptor, here you can observe that they repeated but not acute imipramine treatment induced a decrease both in 𝑃2X7 in red and in 𝑃2X4 receptors in blue here, in the ventral hippocampus of the stressed animals. However, no alterations was observed in the dorsal hippocampus or in frontal cortex. Therefore, this result suggests that the antidepressant response may be associated with a decrease in P2X7 receptor expression in the ventral hippocampus. Based on that, it's plausible to question, what is the effect of the P2X7 receptor inhibition? So, preclinical studies from our group showed for the first time that the treatment with a P2X receptor antagonist, 𝑬𝒑𝒊𝒂𝒏𝒆, reduced the mobility time of animals exposed to the force-field test, which is interpreted as an antidepressant-like effect. Next, the involvement of the nitric oxide pathway was investigated, and in the prefrontal cortex, we could observe that the 𝑬𝒑𝒊𝒂 treatment reduced the NNOS phosphorylation and residue related to its activation, and also reduced the nitrate levels. These results indicate that P2X receptor blockade decreases NNOS activation and nitric oxide levels in prefrontal cortex of stressed animals. And moreover, immunoprecipitation assay indicated that the P2X7 receptor directly interacted with NNOS. Based on that, it was proposed that stress-stimulate ATP release, which can act presynaptically in P2X receptors, induce the glutamate release, as well as the ATP can act in postsynaptic P2X receptors, leading to the increase of nitric oxide production. Increased level of both nitric oxide and glutamate may result in the behavioral consequence of stress. Therefore, blocking this pathway with P2X receptor antagonists may induce an antidepressant effect. And it was shown in this study where the repeated treatment with brilliant blue D, a P2X7 receptor antagonist, more selective than the 𝑬𝒑𝒊𝒂𝒏𝒆, induced a depressant-like effect in the suspension tail test. Corroborating this result, knockout mice for these receptors also presented an antidepressant-like phenotype. Here, these responses were associated to increased levels of noradrenaline in the amygdala and decreased plasma levels of ACT8 and corticosterone. Corroborating these results, we observed that with the repeated treatment, but not acute administration of brilliant blue D, decreased the number of escape failures, but induced no alterations in the trial crossings in the learned helplessness model. As mentioned before, which can be interpreted as an antidepressant-like effect. Lately, Dumas' group showed that an even more selective antagonist for P2X7 receptors, the A804598, which I will call from now on of just A. So the work showed that this drug prevented the decrease in the sucrose preference induced by the exposure of the chronic unpredictable stress model. In the same study, immobilization stress during 6 minutes increased both the ATP and glutamate release and hippocampus microdialysis. Interestingly, the restrained stress also induced an increase in the interleukin-1 beta and TNF alpha, which was prevented by the administration of A, here shown in the blue line. This study suggested that the response induced by stress exposure could be mediated by inflammation activation. Therefore, we have that genetic or pharmacological inhibition of 𝑃2X7 receptors induced depressive-like effect, which may be associated to several mechanisms such as decreased monoamine, nitric oxide, and glutamate levels, as well as diminished neuroimmune response. However, the involvement of BDNF, a neurotrophic factor required for the antidepressant effect of monoaminergic and non-monoaminergic drugs, had never been studied so far. For this reason, we aimed to answer the following question. BDNF signaling pathway is associated with the antidepressant-like effect of 𝑃2X7 receptor antagonists. Talking a little bit about BDNF, this neurotrophin interacts with TrkB receptors, leading to the autophosphorylation of these receptors and activation of two important intracellular cascades, the P13 kinase 2-AQ2 and 𝑧/𝒂𝒃 cascade. Both cascades lead to the mTOR and P70S6 kinase activation, and they are related to neuronal survival, neuroplasticity, protein translation, and also neuronal growth. For this reason, the levels of BDNF, AQT, ERK, mTOR were evaluated on the present study. For that, we used an animal model of depression based on the selective breeding, the Flinders-resistant line, or FRL, and Flinders-sensitive line, or FSL rats. The Flinders-sensitive line are more present, the Flinders-sensitive lines rats presented an increase in the immobility time in the first swim test, even with no pre-exposure to stress. And this behavior may be prevented by the antidepressant treatment. That's why this model could be used as an animal model to study depressive-like behaviors. Moreover, they present another molecular features that resemble the stress consequence. So, we treated the FL rats with daily injections of vehicle during 7 days, and the FSL rats were treated with vehicle or 30 milligrams of A during the same period. 1 hour after the last injections, the animals were exposed to the open field test and right after to the fourth swim test. Next, animals were decapitated and had the frontal cortex, ventral, and dorsal hippocampus dissected for the Western blotting analysis of the aforementioned proteins. Then we observed that the repeated treatment with A, but not acute, decreased the immobility time in the first swim test of the FSL rats. As no alterations were induced in the locomotion, which was evaluated by the open field test, we can interpret this result as an antidepressant-like effect. On ventral hippocampus, FSL rats presented decreased BDNF levels, which was reversed by the treatment with A, and the treatment with A increased the AQT expression the same structure of FSL rats. Moreover, FSL rats presented the lower expression of 𝑃70S6 kinase, which was no more observed after the treatment with A. On dorsal hippocampus, the P2X7 receptor blockade increased the expression of BDNF levels and decreased the expression of P70S6 kinase levels in FSL rats. No alterations on this protein's expression were observed in the frontal cortex. So what we have is that the repeated treatment with A induced an antidepressant-like effect in FSL rats associated with an increase in BDNF-TrkB-AQT signaling in the ventral hippocampus. This result is in line with wild data showing that the antidepressant effect is accompanied by the activation of BDNF signaling pathway in the hippocampus of rodents. Altogether, animal models suggest that the blockade of P2X7 receptors can be a possible strategy to depression treatment. But for sure that clinical studies are required to better elucidate that. So I finished my presentation. Thank you, my PhD supervisors, 𝑧𝒊𝒂𝒏𝒂 and 𝑬𝒍𝒊𝒂. I also thank so much my collaborators, Caroline Biagioni, Plínio Casarotto, and Laura Stanchini, as well as the technicians and other collaborators for Aarhus University and University of São Paulo. Thanks for the funding agents. And thanks for Professor Henny for receiving me in his lab when I'm developing my postdoctoral study investigating the role of purinergic receptors in animal models of Alzheimer's disease in collaboration with the Professor Beatriz Oliveira and Sérgio Ferreira.
So I would then just thank you for your very interesting presentation and would like to introduce the next speaker. The next speaker is Agatha Oliveira Giacomelli. She's graduated in pharmacy and has a master in biochemistry by Federal University of Santa Catarina, PhD in biochemistry by the University of São Paulo. And currently she's a postdoctoral research in the Laboratory of Neuroscience in the Chemistry Institute, working together with Professor Henny. And since her PhD, she has been very much interested and developed results or research in purinergic signaling in all the locomotor chains associated to Parkinson's disease. So thank you, Agatha, please.
Thank you, Jacques, Professor Samia, for your introduction. I would like to thank FeSBE for the opportunity to present my work. And today I will talk with you about the whole of purinergic receptors in Parkinson's disease. And using some data I gathered during my my doctoral degree in the laboratory of Professor Henny Ulrich on the Institute of Chemistry of the University of São Paulo. So briefly, the Parkinson's disease is the second most prevalent neurodegenerative disease. It affects near 1% of people between 65 and 69 years old and can reach 3% of people with more than 80 years old. The main symptoms of Parkinson's disease are motor symptoms that are very characteristic of this disease, like resting tremor, shaking palsy, impaired posture and balance, and bradykinesia. Also, Parkinson's disease patients present some non-motor but neural symptoms as cognitive dysfunctions, sleep disorders, psychosis, and mood disorders. The main characteristic of the pathophysiology of this disease is the death of dopaminergic neurons from the Negro-striatal pathway. Here we have the representation of a human brain and here a murine brain. Here we can see in red the Negro-striatal pathway, where neurons from the substantia nigra projects its ramifications to the striatum. Current treatments for Parkinson's disease are symptomatic, which means there's no known cure for this disease. And the standard treatment is with the compound levodopa, that is L-dopa. L-dopa is a precursor of dopamine, but unlike dopamine, this L-dopa is able to cross the blood-brain barrier and be uptaken by dopaminergic neurons, transformed and released as dopamine. However, long-term usage of this drug induces several collateral effects. For example, dyskinesia, that is a motor collateral effect, very incapacitating, which highlights the need of a better understanding of Parkinson's disease and also the identification of new targets for treatment. So together with the neurodegenerative process that occurs in Parkinson's disease, we have the neuroinflammation process. How this happens? We know that when neurons in neurodegeneration, they release several molecules that can act as danger-associated molecular patterns. That's the dumps. These dumps can act in microglia cells, for example, inducing activation of microglia cells, proliferation of the cells, and recruitment of the cells, also increasing the release of cytokines that can increase the neurodegenerative process. One of these dumps we can mention is the purines ATP, ADP, UTP, and UDP. These molecules are involved in the purinergic signaling. Receptors from this system include SPIR1 receptors that are activated by adenosine, but I will not talk too much about them today because I'll focus on the second part of the purinergic system, that is the P2 receptors. P2 receptors can be subdivided in ionotropic P2X receptors that forms membranous ion channels that allows the flow of sodium, calcium, and potassium, and are ligands for adenosine derivatives, I mean ADP and ATP. And also we have the P2Y receptors that are metabotropic receptors that, besides being able to be activated by adenosine derivatives, can be also activated by uridine derivatives, so like UDP and UGP. There are several P2Y receptors with different holes in physiological process, and they are kind of different with relation to which cells they are expressed and which system they are expressed. One interesting receptors that my colleague Deidiane already spoke about is the P2X7 receptors. This receptor, they have an interesting feature, that is, when they are briefly activated, they can allow a brief depolarization that will induce moderate cytokine release that will influence in inflammation and cell death. However, when this receptor is the activation of these receptors is sustained, it can induce a sustained depolarization that allows the opening of large membranous pores that allow the flow of big molecules that can impair the cellular homeostasis and induce cellular apoptosis. This is the case, for example, when ATP acts as dumps in brain injuries. So in the central nervous system, purinergic signaling can exert several processes. For example, ATP can act as neurotransmitters and can act in the communication between astrocytes and neurons, and between neurons. Also, as I already mentioned, purines can act as dumps in in a neurodegenerative and inflammatory process, inducing, for example, the activation of microglia cells. So the main question that I made in my doctoral research was which purinergic receptors are involved in Parkinson's disease. So to answer this question, first, I induced an animal model of Parkinson's disease. I made a screening of P2 receptors to try to identify possible targets for the disease. So I selected two targets, two purinergic receptors targets, to induce an in vivo modulation in this animal model of Parkinson's disease. To this purpose, I used male sprague-Dawley rats, and I injected the compound 6-hydroxydopamine. This compound is an analog of dopamine that can be transported to dopaminergic neurons by dopamine transporters, and once on these neurons, it can block the complex 1 of the electron transport chain and induce the production of reactive oxygen species and cell death. So I injected this compound, and after 1, 3, or 5 weeks, I collected samples to analyze the P2 receptors gene expression. And this is what I got for P2X receptors. As we can see here, the most interesting, here we have in white, we have the control hemisphere, the intact hemisphere, and here in gray or in red, in this case, we have the injured hemispheres, 1, 3, or 5 weeks after 6-hydroxydopamine injection. So we, as expected, we saw an interesting results for P2X7 receptors. As we can see here, we have an increase on P2X7 receptor gene expression during these weeks. And this could be because of the property of these receptors of opening big pores during brain injury due to large ATP release. For P2Y receptors, what we saw was interesting results for the P2Y6 receptors. As we can see here, we have both in control and in injured hemisphere an increase on gene expression of these receptors. So we decided to analyze more deeply this receptor. At this time, work came out showing that P2Y6 gene expression was increased in peripheral blood mononuclear cells of Parkinson's disease patients. And the author suggested that this could be a biological marker for Parkinson's disease due to the neuroinflammation that is occurring in the brain of these patients. So after I selected these two targets, we proceeded to the modulation in vivo. So for the P2X7 receptors, we decided to use an antagonist that's the brilliant blue G that I will call BBG. BBG is a blue food dye that's widely used, for example, in M&Ms and in Gatorade. And to study the effect of this antagonist, we injected 6-hydroxydopamine, and after 1 week, we injected BBG for 7 days daily, and in the 8th day, we collected samples for analysis. For P2Y6 receptors, we decided to go with prevention protocol, which means that we first injected the antagonist of P2Y6 receptors, and then we injected the compound to induce the animal model of Parkinson's disease. That is the opposite of what we made for P2X7, when we first induced the model and then treated the animals. So the P2Y6 receptor antagonist MRS-2578 was injected in striatum, and 1 week later, then we administered the 6-hydroxydopamine. 1 week later, we made the analysis. The data I'm going to show you today is regarding the immunohistochemistry data that I got from these results. For these experiments, we stained dopaminergic neurons with tyrosine hydroxylase, for tyrosine hydroxylase that is an enzyme involved in the synthesis of dopamine. And also we analyzed EBA1, that is a microglial marker, to analyze microglial morphology in these animals. So we used two brain areas. First, as I mentioned, the substantia nigra, where the cell bodies of the dopaminergic neurons are located. So we counted the cell numbers. And also we used the region of the striatum, that is where only ramifications are present. So in this area, we analyze optical density. So in the striatum of this, the BBG animals, the antagonists of 𝑃2X7 receptor administration, we can see here we have in this column the injured hemisphere of these animals, and this column we have the intact hemisphere of these animals. We treated with 4 different doses of BBG, and what we got is that at the dose of 75 milligrams per kilogram, BBG was able to restore dopamine in the injured hemisphere. And when we look at the substantia nigra, what we can see, same here, this is the injured site and this is the control site, we can see that not only the number of dopaminergic neurons were restored at BBG at the dose of 75 milligrams per kilogram, but also at the dose of 50 milligrams per kilogram. Actually, we had at this moment already published this data, previously published this data, but in the work that I'm presenting to you today, we decided to go with a higher dose to see if we got better results. And actually, we could see also a difference in the striatum, as I already showed you. So the protocol, the data that I just showed you, and the ones that we published with BBG at the dose of 50 milligrams per kilograms are the only ones in the literature that show a neuroregenerative effect of BBG treatment. In the literature, we can find other studies using 𝑃2X7 receptors antagonists, but these other works used a prevention protocol, which means they treated with BBG before induced the lesion. When we look at microglia cells in these animals treated with BBG, we can see that here we have injured hemisphere, and here we have control hemisphere of animals treated with vehicle, that is saline, or with BBG. And we can't see any difference between any samples. But when we look at the substantia nigra, we can clearly here see that we have an increase in EBA1 marker that relates with, that indicates that there's a shift in microglial phenotype for activated microglia. And when we treated these animals with BBG, we can see a reduction in this marker, indicating that BBG could be acting modulating microglia activation as we proposed. So how do we think BBG, our model, is working? So we have the degenerating neurons that are undergoing the degeneration because of the 6-hydroxydopamine. And these neurons, they induce a massive ATP release. This ATP could be acting in 𝑃2X7 receptors, in astrocytes, or in microglia cells, inducing the release of cytokines and ROS that could be inducing cell death. Could be acting also in oligodendrocytes, inducing demyelination. And although it is still controversial whether or not neurons express 𝑃2X7 receptors, this ATP could be also acting in 𝑃2X7 receptors of this cell type. When we take a closer look at the microglia cells, we can see that there is known that 𝑃2X7 receptor hyperactivation by this ATP induce activation of microglia cells and proliferation. And this active microglia release chemokines that are gonna recruit other microglia cells. And also they induce the release of cytokines that can activate more microglia cells or can induce more cell death that are gonna release more ATP, inducing an expansion of the lesion and the worsening of the scenario. So BBG could be acting in two fronts. First, by modulating inflammatory milieu, by, for example, modulating microglia activation and preventing 𝑃2X7 receptor pore formation and impaired cellular homeostasis. But also, BBG could be acting by increasing neurogenesis. This hypothesis is based on that was demonstrated that in the neurogenic areas of the adult brain, the inhibition, the downregulation of 𝑃2X7 receptors was essential to the neuronal differentiation of neuroblasts. So BBG could be inducing this differentiation in neurons to repopulate the injured area. When we look at the results regarding the P2Y6 receptor antagonist, what we can see here in the injured hemisphere, in the control hemisphere, we can't see any difference for TH staining, which means there's no protection in the striatum deficit of dopamine ramifications. However, when we look at substantia nigra, we can see here the injured hemisphere control. We have the injured hemisphere of animals treated with MRS-2578. We can see that this compound was able to partially protect these neurons from death induced by 6-hydroxydopamine. And when we look at microglia cells, as we observed for BBG, there is no difference between samples in the striatum. However, when we look again at the substantia nigra, we can clearly see that there is a microglia activation in the injured hemispheres of animals without treatment. And this microglia activation can be partially prevented by the administration of the P2Y6 receptor antagonist, suggesting that P2Y6 antagonists could be acting in this in microglia cells as well. So, which are the functions of P2Y6 receptors in the central nervous system? It was shown, for example, that here we have in green we have dopaminergic neurons, and in red we have P2Y6 marker. It was demonstrated that dopaminergic neurons from the substantia nigra express P2Y6 receptor. However, the main function of P2Y6 receptors in the central nervous system is regarding microglia cells. As we can see here, for example, microglia cells during the process of activation, microglia cells downregulate the expression of other purinergic receptors that are the P2Y12 receptors, while they present an increase in the P2Y6 receptor expression. The P2Y12 receptors is really related to the migration process of microglia cells, while the P2Y6 receptors are involved in the phagocytosis process of microglia cells. So, in vitro studies showed that activation of microglia cells induced by LPS induces an increase in P2Y6 receptor expression, along with an increase in cytokine release. And this effect was accompanied by an increase in the phagocytosis process. But another interesting effect was observed when activated by LPS in co-culture with neurons. The microglia cells also present a process called phagoptosis. In this process, microglia cells phagocytes viable neurons, not neurons undergoing cell death. So this could be a possible mechanism that this effect was abolished in this study by MRS-2578, and this could be a possible mechanism for the P2Y6 receptor antagonism affecting our animal model of Parkinson's disease. So altogether, we have that neurodegenerative process induce ATP release that could be activating microglial cells, inducing proliferation and recruitment that are gonna increase release of cytokines and induce cell death. And moreover, that these molecules, DAMPs molecules, could be stressing viable neurons that release UGP and activate P2Y6 receptors in microglia cells that induce the process of phagocytosis, both mechanisms increasing injury expansion and worsening. And our treatment could be with P2Y6 receptor antagonists, could be by preventing the phagocytosis process in microglia cells, while our treatment with BBG could be preventing microglia activation. Also, BBG could be inducing the neuronal differentiation of neuroblast cell to repopulate the injured site. As perspectives, we want to now analyze the effect of these two receptors in the alpha-synuclein model. Alpha-synuclein is a in Parkinson's disease.
Just 1 second. Sorry to interrupt, but we are actually running out of time. So maybe if you can, one more minute. Yeah, okay, thank you.
So the alpha-synuclein model induced the ATP release and microglia cells that could be important for our study. Just for you to know, there is other purinergic receptors that are intensely studied. For example, P2Y2 receptors that are antagonized by caffeine that are still a hot topic, and P2X1 receptors that are shown to be involved in accumulation of alpha-synuclein. And I would like to thank my former colleagues and my actual colleagues that work with me in this in my doctoral degree, and all my other colleagues of the lab and the lab of Claudiana Lameu, professor. And also I would like to thank my colleagues that are working with Parkinson's disease currently. So thank you and sorry for the time.
Thank you, Agatha, for this brilliant presentation, very interesting results. As I said, we leave the questions for the end of all the presentations. And for the sake of time, then now I would like to introduce the next speaker, Talita Glaser. She will speak about purinergic signaling in Huntington's disease. And Talita is graduated at the University of São Paulo with research experience with stem cells, and she did her PhD under the supervision of Professor Henning with exchange or sandwich PhD with 𝑧 at Kyoto in Japan, and she also did part of her postdoctoral training in Harvard Medical School in the United States. So thank you, Talita, and please you can start.
I'd like to thank everyone for inviting me to give this speak. It's a pleasure, and I'm very grateful to have the opportunity to show part of my postdoc research. So during my PhD, I've worked with embryonic stem cells, calcium signaling, and biological, like a developmental cell clock. I did this, as Professor Samia mentioned, a bit in Japan, and of course with Professor Henning, which I have been working since 2010. First of all, I'm going to talk a bit about Huntington's disease. Many of you may not know about this disease, or know very few. Some have just heard of it in the TV series House, Dr. House. So Huntington's disease is a disease that was first described in 1872 by George Huntington, which was just 22 years old. First of all, the name he gave was like hereditary chorea. Chorea is the word that comes from like the Latin and Greek chorus, or group of dance. And with, like, for example, choreography, this word, like the person can stop moving. The involuntary movements are not inhibited. So the person can't stop shaking and moving. Different from Parkinson, which Agatha just told, which usually like the person has difficulties to induce the movement. In Huntington, the person can't stop moving. And for example, Huntington disease has been described the gene which characterizes the mutation in the gene that characterizes the disease. So this is first of all called as a neurodegenerative disease. It's a dominant disease, so one allele mutated is enough to inherit this disease and have the symptoms. It has the loss of GABAergic neurons from the basal ganglia. It's a rare disease, so like 4 to 15 cases in 100,000 citizens. The lifespans are 10 to 20 years after diagnosis, which is usually around 30 to 40 years old. This is the later onset. There's also a different type in which affect kids from 7 years old on. It has many similar symptoms as Parkinson's, as Agatha just mentioned. For example, because it's a motor disease, so we have problems like impaired cognitive skills, we have like involuntary movements, difficulty in choosing the right words like dysarthria, muscle stiffness, which is called hypertonia. However, it's not very well studied yet, but it's very clear that these people, these patients, they develop personality change and mood behavior change. Like, for example, the person was first a very calm and relaxed person, suddenly become very aggressive and very impatient. Also, the person developed psychosis, mainly in young ones. And also depression, like around 10% of the patients die of suicide. Around 36% of the patients die with choking. So they try to swallow and they die. It's a very sad way to die. And these people, they need full-time care. So they very young ones, they're at stop being labor force, so they can't contribute much to society. Here, just some pictures to exemplify, to show what happens in the brain of some patients of Huntington. So here we have the normal brain, the healthy one, and around here, the ventricles, we can see basal ganglia. In the case of a person with Huntington disease, we have a huge atrophy of the basal ganglia, but not just the basal ganglia, but the cortex itself also decreased. As we can see here clearly in the post-mortem brains, the cortex is already degenerate. And here's an MRI. It's clear to see the difference between someone with Huntington and a healthy one. So this huge damage in the brain, very young, like imagine like some kids with 7 years old can already develop this disease. So even though it's rare, it's so aggressive. So, and we still don't have any cure for it. So just in 1993, they find out which gene is mutated and can cause this disease. However, they still don't know the biology and how this mutation can lead to disease, even though there's a huge amount of researchers and many groups studying on it. The gene is called Huntingtin or IT15, and it's characterized by in the N terminal of the protein, we have a stretch of many repetitions of CAG, which encodes glutamine. So we call it polyQ. If you have over 39, so with 40 repetitions of glutamine, the person will develop the Huntington disease symptoms. The Huntingtin, the wild type one, the healthy one, they are important. We know now that for maintenance of neurons, for the embryonic development, for chemical signaling, for example, for the release of neurotransmitters, for the transportation of materials within the cells, like even the organelles can be trafficked.
connection between the proteins, like it's as known as a scaffold protein. So it can connect, like, for example, receptors with other receptors to make some specialized pools of receptors in the membrane for neurotransmission. And also it has an anti-apoptotic property. However, when we have this mutation that stretch in the polyQ section in the N terminal, the mutant protein aggregates, and this can make get some new functions which this protein did not have before. For example, it binds to many transcription factors and other signaling molecules, and they can dysregulate transcription of many important genes. It can abolish the trafficking, for example, of BDNF, which Deidiane already mentioned to us before in her presentation, with a neurotrophic factor. It's very important for the brain maintenance. And also it can increase, exacerbate calcium signaling, which can trigger depolarize the membrane of mitochondria, release cytochrome C, and induce apoptosis of the cells. Recently, there's a new shift in the Huntington's disease paradigms, which is like, it's not just a neurodegenerative disease, but also it's a neurodevelopmental disease. So early on in development, this disease already being set on. So in 2008, with an alignment analysis, showed that polyQ is a synapomorphy of deuterostomia. What are deuterostomia? It's like during the early in the development of the embryo. So we have, for example, fertilization, the morula, the blastula, and when the embryo, for example, the mammal one, get implanted in the womb, we start to have the production of the 3 germ layers. And then after these 3 germ layers formation, the embryo tissue folds to make the whole fetus. So the first hole, the first cavity made as in deuterostomia is the anus, is the anal. So this happens since echinodermata, which is starfish, and this is very important in evolution, because the folding, the gastrulation is different since we have this polyQ appearing in this Huntington gene. Also, a higher amount of glutamine repetitions, like longer is the stretch of polyQ, more developed, more complex is the central nervous system. In addition, the knockout, like the removal of this Huntington, the wild type, is lethal. We don't have gastrulation. So this is a very important protein for the embryo development. And as I mentioned before, over 72 repetitions of glutamine, you can have a kid developing Huntington's disease with 7 years old. Purinergic signaling, like Agatha already explained about, it's activated by ATP, ADP, or even adenosine. We focus on, in this work, in P2 receptors, which are activated by ATP. It's been described that purinergic signaling is present in embryo development very early on. Being expressed, it was showed by Zimmerman in 2006 already. And if you interfere in the signaling by using antagonists during gastrulation, in the early gastrulation, the embryo doesn't develop head, trunk, somites, notochord, like it can't be developed. If in the midway, the embryos don't have heads, but they still have trunks and tails. And if it's the end of gastrulation, the embryo can develop some. So we think that Huntington and purinergic are very well correlated. And not only this, but Make and Nedergaard, he already showed that you need downregulation of purinergic signaling along neuronal differentiation, and the neural precursor cells are themselves the source of ATP. As Dr. Henning showed yesterday, like just to gather the data, so to explain a bit, introduce to who that didn't watch his presentation, we observe spontaneous calcium oscillations, which the cells are doing. We're not triggering with any molecule. It's just like the cells in the media along differentiation of embryonic stem cells. So here on the right, we can see this signaling being triggered, the influx of calcium. We show that ATP is higher concentration in the extracellular milieu in the neural precursor stage. We can differentiate cells into neurons very well. And during this process of differentiation, we had two types of oscillations we called spikes and waves. And in the data of the paper we published recently in 2000, now this year in April, in molecular psychiatry, we showed that P2Y2 receptors, they can favor GABAergic differentiation, controlling the cell clock. So the combination between P2Y2, P2X7, and voltage-gated calcium channels, they can trigger spikes, oscillation, or waves in a different way, controlling the transcription of some transcription factors that are pro-neuronal, like ASL1 and neurogenin 2, and the pattern of oscillating of SL1 to a stable one. And if it's increased, we can make like a we called neurogenic index. If we treated cells with 2-TP, with an agonist of P2Y2 receptors, we can have this much increaser NASH1 over neurogenin 2, and this was enough to increase to favor GABAergic differentiation. So, as Henning mentioned yesterday, P2Y2 receptor increases spikes, oscillations, which can control SL1 expression, which leads to GABAergic neurons differentiation. Which are the neurons that die in Huntington disease? So what about, our question is, what about P2Y2 receptor and the calcium oscillations in Huntington disease? Would they be participating in it? Would they be related to Huntington and the neurodevelopment? And they're related to the new paradigm shift? So using in induced pluripotent stem cells, which Stephen Henning already presented 2 days ago, which are cells that are removed from the patient, the adult one. It can be from the dental pulp, it can be from the blood, from the urine, from the skin. In this case, we use cells that were from the skin of the patients of Huntington. It was reprogrammed, and then these cells were differentiated to neural cells. And what we observe is that the neural precursor cells of these patients, they already show increased basal calcium. So the calcium homeostasis is affected already, it's impaired. And not only this, the spontaneous oscillations, the calcium ones, they have two types, the spikes and waves. We can see here in the control, and so this darker one is a spike and the lighter one is wave. And in Huntington, we just observed waves, but we couldn't observe spikes. Spikes were like tonic, very short, so we can detect it. So it shows that there's already a difference between Huntington and wild type ones, healthy ones, early on, since the neural precursor cells are being established in the central nervous system. Oops. Also, we observed that these cells, they were more like sensitive to ATP challenge. So they had a calcium response that was higher when we induced it with the ATP. However, after 10 days of differentiation for GABAergic neuron, the Huntington cells, they can't have, they don't show any calcium influx anymore, while the wild type one has. Not only this, we also showed that with transfection of this of a wild type 1 with 23 repetitions of polyQ, or with 74 is the disease, 23 is healthy. The cells, they can respond to BZ-ATP, but they can't be responsive, or they have lower response to the P2Y2 agonist. And the cells of these patients, they express much higher levels of P2Y2. We think it's kind of feedback, like the cells are trying to compensate the low response of the receptor by increasing its expression there, its mRNA. Because it seems that this P2Y2 receptor is not just important for the differentiation, but also to the maintenance and survival of the cells. Because when it's is decreased, the cells trying to compensate it. Also, we use mouse embryonic stem cells, not just iPS cells, because we know that iPS cells can have huge variability and among patients and clones. So we also did a genetic model using mouse embryonic stem cells. And so the cells of embryonic cells were differentiated to GABAergic neurons in vitro. So they express GAD65, 67. We've made the cell lines first with knockout. We removed the Huntington gene, and then we induce the expression of we overexpress the wild type or the mutant one. So we had an empty, we can say like empty background, and then we add to express our gene of interest. It's just the N-terminal part. And we observed that the cells that overexpress the mutant Huntington, they differentiated faster. We had increased rates of differentiation. And of course, because of that, we have a small amount of precursor cells in a pool of precursor cells, which goes according with some data in the literature with transgenic mice of models of Huntington, which shows that Huntington animals, the adult ones, they show decreased pools of neuroprecursor cells in the neurogenic areas in the brain. So we see here that even in the beginning of the development, we already have a shorter, decreased amount of precursor cells in the brain to be later on used for adult neurogenesis. We could explain by around the onset, around 30 to 40 years old, which when our neurogenesis decreased a lot. That's why we learn less when we're achieving like 30 to 4 years old. Also, we start to not listen too well, like we start to lose a bit of our senses like smell, listening.
Talita, sorry just to interrupt, but it's just to say, yeah, you have to.
Yeah, it's finished.
It's finished. Okay.
Yeah. And we reproduced this thing in mouse embryonic stem cells. We did the same. We differentiated them. These ones that are blank background, and we see also P2Y2 was low responsive, even though it was being expressed. We didn't see difference here, but they were still expressing this receptor. So our conclusion is that the P2Y2 controls calcium spikes. These receptors response is impaired in Huntington disease, precursor cells, and GABAergic neurons. And they decrease, because of that, it's decreased the storage of precursor cells in adult for neurogenesis. And I'd like to thank people from Japan, from Harvard, my members in the lab, and also some from Claudiana's lab. They also give me a lot of support to do this work. And also the technicians, Denise and Zita. Thank you very much.
Thank you, Talita, for this very interesting presentation. So I would like to invite the next speaker, and the next speaker is Juçiana Azambuja. She will talk about adenosine production in tumor microenvironment as a target for cancer therapy. Juçiana is from the south of Brazil. She did her graduation and master in Federal University of Pelotas. And since then, she has been interested in brain cancer and the relationship between inflammation and progression of tumors, and more recently, the involvement of the purinergic signaling as a target for treating these conditions. Currently, I forgot to mention, Juliana is working at Unicamp.
I'm going to talk about adenosine production in the tumor microenvironment as a target for cancer therapy. So today I'm talking about glioma or glioblastoma. So glioma is a kind of brain tumor derived from astrocytes. And glioma is not the most prevalent and incident type of cancer, but is a very dangerous and aggressive type of cancer. So the treatment is made based on surgery when it's possible, followed by radiotherapy and or chemotherapy. But glioblastoma is still a big challenge for us to treat because of the location, the complexity of the brain, and also the presence of the BBB, which decrease the options for treat glioblastoma patients. In this situation, also glioma is a very unstable kind of tumor, and at least 90% of the brain tumors developed chemoresistance, which means 90% of the patients, the therapy stopped working for them, and they're led without options for treatment. So in this situation, glioma now is the third leading case of death among Caucasian patients, and also just 5% of the patients are alive 5 years after the diagnosis. Also, the median of survival is just 12 months. So we have to choose new targets for improve this situation, and this is urgent. So how my colleagues told you today, purinergic signaling is coming out in the past years as a target for several diseases, several brain diseases, and also for cancer. So in cancer, what we know is several cancer types and also brain tumors, they increase the expression of one enzyme, CD73, which is an enzyme present in the cell membrane and is the main source of adenosine in the tumor microenvironment. So this adenosine has several effects. So when adenosine binds adenosine receptors in cancer cells, this leads to increase in invasion, metastasis, proliferation, chemoresistance, migration, and angiogenesis. But CD73 and adenosine, they are coming out as important players in the tumor microenvironment because they have effects not just in the tumor cells, but also in other cells present in the tumor microenvironment. So what we know is that tumor cells, they recruited immune cells for the tumor microenvironment. And these immune cells, when they reach there, they should work attacking the tumor cells and killing them as a cell with a problem and controlling tumor progression. But often what happens is that tumor cells, they are recruited for the tumor microenvironment, and when they arrive there, they are educated by tumor cells to assume an immunosuppressive and pro-tumor phenotype. And who plays an important role in this situation is adenosine. So adenosine works as a brake for immune cells and stop these anti-tumor effects and create an immunosuppressive and induce tumor proliferation. So in this scenario, our question is, could CD73 and adenosine production be a target for cancer therapy and for brain tumors therapy? So we start evaluating the effects of CD73 and adenosine in glioma cell biology. For this, we first treat the glioma cells with adenosine and ANP, and we evaluated the cell viability. As you can see in the top graph, adenosine increased glioma cell viability in low concentrations between 0.1 micromolar and 50 micromolar. And also in the opposite side, ANP have opposite effects, decreasing glioblastoma cell viability. So this data made us create a hypothesis about if we block CD73, we can prevent adenosine production in the extracellular space and maybe control tumor progression. So in order to evaluate this, we worked with two experimental conditions. The first one is the treatment of cells with a CD73 pharmacological inhibitor, APCP. And the second condition is knockdown CD73 with new sequences for with siRNA for CD73. So you worked with glioma cell lines in vitro. We created these two conditions and we evaluated some glioma cell biology behaviors. Also, in vivo, we evaluated the tumor progression. So we designed these 4 sequences. One sequence is the siRNA scramble, which our control is not supposed to do anything with the cells. And we have 2 sequences, siRNA for CD73 in rats, and 1 sequence for CD73 in humans. So our first step was evaluated the efficiency of the sequences in knockdown CD73. And as you can see here, all sequences, they are efficient in decrease the CD73 expression and CD73 activity in glioma human and white cells in vitro. And this efficiency is more than 90%. So after that, we evaluated the effects of this CD73 block in vitro in glioma cells. As you can see here, APCP, which is the CD73 inhibitor, it decreases glioma proliferation in vitro in a dose-dependent way in 30%. Also, when we knock down CD73, we also decrease glioma proliferation in 40%. In the sequence, when we knock down CD73, and after we treat the cells with adenosine, we revert partial or total these effects, showing that CD73 controls glioblastoma proliferation, and this is mediated by adenosine. And also, when we block this enzyme, we are able to reduce glioblastoma proliferation. After looking for chemoresistance, we treat 2 glioblastoma cell lines with temozolomide, the gold standard for glioblastoma treatment, and nothing changed regarding cell viability in these cells. But when we worked with a combined therapy, blocking CD73 and also in the same time treating with temozolomide, we are able to reduce glioma cell viability. Also, we reduce EC50 for this drug, showing that CD73 is also a good target for make the glioma cells start work answer again for the gold standard treatment temozolomide. And maybe a combined therapy these two strategies could be a good way to treat the patients with developing chemoresistance. Also, we looked for migration and invasion of the cells after the treatment. In the first, we realized that adenosine alone increased glioblastoma migration and invasion, and this is related with increase in NP2 and vimentin expression on these cells. In addition, when we block CD73 expression or when we knock down the enzyme, we have the opposite effects. We decrease migration and invasion in 50% in these cells, and this is related with decrease in NP2 and vimentin expression. One more time, when we knock down the enzyme and after add adenosine in the cells, we revert these effects, showing that adenosine and CD73 control glioma migration and invasion. This is mediated by adenosine production, and block CD73 is a good way to impair this migration and invasion in cancer cells. After we worked with in vivo 6/6 immunocompetent glioblastoma model. In this model, in the day 1, we injected glioblastoma cell line in the brain of the rats. After 7 days, in the day 7, 14, and 21, we treated animals via ICV. And in the day 23, we made euthanasia and we looked at the tumors. In the pictures and in the graph, you can see the tumor size after the treatment. And CD73 block with APCP or with the siRNA reduced tumor size also in vivo in 40 and 45%. Also, we looked at adenosine levels in the brain of these animals, and we showed that treatment with APCP or siRNA decreased adenosine levels in the brain in 90 or 95%, showing that this approach was efficient in block CD73 also in vivo. And only in the group which we knocked down CD73, we also showed some increase in leukocyte infiltration. So maybe this increase in leukocyte infiltration is related with the presence of the CD73 in the membrane and not just mediated by adenosine production. So in this first data, we showed that glioblastoma, they express CD73. We are able to design new sequences, efficient knockdown CD73 and decrease adenosine production. This knockdown of CD73 is related with increase in temozolomide sensibility, decrease in tumor malignity, decrease in tumor proliferation, and decrease in migration. Also, in vivo, block of CD73 was able to reduce the tumor size and induce tumor regression without showing any sign of damaging. We also realized that in the group which we knocked down CD73, we have an increase in NF-κB pathway and an increase in leukocyte infiltration, showing that the protein is also important for the CD73 activities. So after that, we concluded that CD73 is a good target for glioblastoma treatment. But as I told you before, glioblastoma is still a challenge for treatment because of the location and because of the BBB. It's hard to design a drug which can reach the brain. And also, siRNA in vivo is not stable when administered alone, naked, have a short half-life. So in order to make it possible to use this approach with siRNA also in patients, we decided design a system for delivering these sequences. We choose to work with a cationic nanoemulsion, which is a lipid-based system. and have a positive charge. This system interacted with the negative charge donated by the siRNA, forming a complex. So this complex is a promising approach for in vivo treatment because it protected the sequence for degradation, increased the delivery in the tumor site in the brain, and also avoid some adverse effects. And this complex could be a new approach to give a new therapeutic options for treat brain diseases, like brain tumors. Also, in the past experiment, we worked with ECV way to administrate the compounds. Here, we choose this complex because they are able to deliver these sequences via nasal route. And nasal route is a good way to deliver therapeutic approach for the brain because I have a connection with the brain and increase the effects in the delivery in the tumor site and also avoid some adverse effects. And more important, is a very comfortable way to administrate compounds for patients where they can make self-administration at home, avoiding hospital internalizations and avoid infections. So now our objective was developed and characterize these complexes, this non-emotion together with siRNA. And in this, we work with glioma cell lines again, and also primary astrocyte cultures. We knock down CD73, but this time using our system, the non-emotion, and we characterize the system and also look at glioblastoma cell behavior in vitro. And one more time, we also look at tumor progression in vivo. So, as first data, we developed a system with good qualities, a site compatible with non-emotions, around 200 nanometers, a polydispersive index close to 0.1 with indicated stability, and as expected, a positive potential beta. Also, you can see the cellular uptake for the complexes in the figure, where in red you can see the non-emotions, and they are highly internalized by the glioma cells. So regarding the CD73 activity, in pink you can see the activity after the treatment with the complex, and the complex are able to knock down CD73 in vitro and reduce CD73 activity in 80%. Also, about the cell viability, the complex are able to reduce the glioblastoma cell viability in 50%, one more time indicating that CD73 is a good target for cancer treatment. And now we also treated astrocytes with our healthy cells from the brain with the same complex. And we observed no alterations in the cell viability, showing that targeting CD73 is selective for reduce the viability of tumor cells, and also indicating that treatment may be safe for in vivo administration. Again, one more time, we worked with in vivo glioma immunocompetent model, where the animals were they went to surgery in the day 1. In the day 5, we started treatment. We treated the animals for 15 days, twice a day via nasal. And in the day 20, we made an euthanasia and tumor analysis. So, as you can see in the graph and in the pictures, the treatment with the complex, they reduce the tumor size in 60% in vivo. Also, as you can see here, the immunohistochemistry in green, CD73 is a highly expressed enzyme for tumors, for brain tumors, and our complex are able to decrease CD73 expression also in vivo via nasal treatment. In red, you can see the neoangiogenesis, CD31. And one more time, the treatment with blocking CD73 was able to decrease angiogenesis in vivo. Here you can see the adenosine levels and ATP, ADP levels in the brain of the rats after the treatment. In D, you can see the adenosine levels. And one more time, we show that the complex are able to reduce adenosine levels in 95% in vivo, showing that treatment was able to knock down the enzyme after nasal administration. After we started comparing our treatment with the gold standard using in the past decades for glioblastoma, TMZ or temozolomide, as I told you before. So you can see that temozolomide reduced the tumor size, of course, as expected, in 30%, 35%. But when we use our approach, blocking CD73 with nano system for delivery, that we also decrease the tumor size, but in between 60 and 80%, which means our treatment, our system, our new approach have better effects when you compare to the gold standard using in the past decades for treat glioblastoma patients. So in the next picture, you can also see a strategy combining non-emotional sRNA and CD73. And what you see is the treatment is able to reduce the tumor size in 85%. We are not able to observe any synergic or additive effect, but we also know now that a combined strategy is not leading to a decrease in the potential of our complex to treat glioblastoma in vivo. And in the last graph, you can also see the adenosine levels. And one more time, adenosine levels were reduced by the complex in the brain of the rats, confirming the CD73 knockdown. So in this, after this, we can conclude that we were able to develop a cationic nanoemulsion which is efficient in complex siRNA for CD73. And this nanoemulsion after nasal administration in rats is able to deliver the siRNA for the brain in the rats and reduce tumor size without showing any damage for the animals. So we knew CD73 is a good target. Now we also have a new approach for delivering this strategy in vivo for brain diseases. So as I told you before, adenosine is not just important for tumors, but it's also very important for the tumor microenvironment. So
Juliana, just sorry to interrupt. We have just maybe 2 more minutes. Then
I'm close to the end. So we look for the immune cells infiltration in this tumor site. So we show that block CD73 is able to reduce macrophages, microglia, and Tregs in the tumor microenvironment. And also CD73 is decreased not just in tumors, but also in macrophages and microglia. So as a conclusion, we developed a system for block CD73 in vivo, which were able to reduce the tumor size. This is related with increase in the different cytokine release profile, with increase in EL6, CCL17, and CCL22, and decrease in inhibitory immune cells related to tumor progression, such microglia, Tregs, and macrophages. So our hypothesis here is that decreasing these inhibitory immune cells and increasing cytokines is able to remove the brake in the immune system, and these immune cells now are able to attack the tumor cells and reduce the tumor progression. So I would like to say thank you for everyone who made this project possible, and thank you for everyone who's listening right now.
Thank you, everyone, and thank you for the brilliant young scientists that presented today and did this very nice job.
I would like also to thank in the name of the 𝑧𝒂𝒂 𝑑𝒂, yes, for accepting our or suggestions of the symposium. Yes, and we are very proud and very content to make part of FeSBE, one of the societies of FeSBE. Yes, and I think that FeSBE has a very important function in experimental science. Yes. So I would like to congratulate the organizer for this. Yes. And again, of course, all the researchers who presented today. It's excellent. Thank you very much.
Thank you very much again. Yeah, thank you, the organization and everyone.